Yes, great. Thank you.
Wonderful. Okay, great. I will share my screen now and then tell you the work that we've been doing on Eterna and at Stanford University and how we've responded in response to the COVID-19 pandemic. So first, thanks so much for having me. It's an honor to be here, and it's so fun to engage with the other side of the globe. My name is Taeseun Kim. I'm from the DOS lab at Stanford University in the biochemistry department. I'm super thrilled to be here, and I'm here to tell you about our initiative, the OpenVaccine Challenge on Eterna, where we have taken on this project to design stabilized mRNA vaccines in response to COVID-19. And the unique way we're doing this through crowdsourcing and through citizen science. So mRNA vaccines, we believe, are one of the keys to addressing this terrible pandemic. And one of the reasons that they're such great, promising platforms is that they are, let me make sure I have a screen. They're incredibly quick to be developed in response to infectious diseases such as COVID-19. So they were the first vaccines that went into clinical trials. Currently, there are over 16 mRNA vaccine efforts underway. There's two notable sort of front runners that have moved on to phase 3 clinical trials and may be close in being approved for use. That is most predominant in the US and in Europe. And the way this works from a scientific perspective is we utilize this key molecule, messenger RNA, that gets translated into an immunogenic protein, which means a protein that can elicit a response from the human body, which is a specific immune response against a specific disease. So in the case of COVID-19, an mRNA molecule would be made into a protein such as the SARS-CoV-2 spike protein, which your body can learn to recognize and develop immunity to. However, there's still challenges with it being such a new platform. So those familiar with the platform may know that there's still many biological unknowns of what happens when you inject mRNA into foreign mRNA into human bodies, how the body will respond, how wide the response is. These are still questions that are certainly being answered and answered at a very high rate because of necessity, but still many unknowns that are being sort of uncovered through these clinical trials. What we see as a really significant challenge for worldwide immunization, which is what you would need for something like COVID-19, is that mRNA vaccines have been known to have poor stability in prefilled syringes compared to traditional protein counterparts. And what we mean by that, I'll share with you briefly. So vaccines, if you've ever gotten a flu vaccine or other vaccines that may be prevalent, they're shipped in syringes that already have the molecule inside of it and can be directly injected into the patient without the need for complex formulation. With mRNA vaccines, however, over time, over certain periods of time and with heat that gets introduced into these syringes through the supply chain when it's being delivered, these mRNA molecules depicted in red here tend to get degraded. And this happens via spontaneous chemical hydrolysis, which we think is a significant challenge to immunizing the world. So you may have read on the news that if you have enough negative 80 degrees Celsius or negative 20 degrees Celsius freezers, you can get these vaccines to the places they need to go. But if you are aiming for worldwide immunization to places that are not as established in supply chain, or especially cold supply chain, as we call it, this sort of challenge depicted in this graphic becomes quite significant in our ability to immunize the world against diseases like COVID-19. So the scientific hypothesis behind what we're doing comes from the fact that RNA are made up of RNA, the central molecule and polymer that we're working with, are made from 4 building blocks, A, C, G, and U. but they can form incredibly different shapes. So on the right, what I've shown here with my colleague Hannah Wayman-Steele of the DOS Lab are all the different shapes that viral genomes can take. So these are viruses whose genomes are made of RNA. And you can see that there are many different shapes, almost like paintings, if you will, that can be made from just these polymers consisting of 4 very basic bases. So what we know from nature is that RNA molecules that are translated, like the genomes of these viruses, can take on many different shapes. And thus, their shapes can be manipulated by changing the bases A, C, G, and U. So our hypothesis behind our project was, can we stabilize mRNA molecules through design? Meaning, if the mRNA molecule or vaccine in this case has this sort of loose shape where depicted in yellow and visually hopefully obvious, yellow denotes single-stranded regions of mRNA. Can we design the mRNA such that we can encourage base pairing and thus sort of closing up these vulnerable loops that may be prone to cutting? So the central hypothesis is that double-stranded mRNA is most likely advantageous for stability. And can we redesign these long mRNA molecules to be more compact and more protected against degradation by switching these 4 very individual bases, A, C, G, and U, to take on more advantageous shapes? And then comes the challenge as a researcher for us to ask, how can we do this on a very, very, very fast time point? So for those of you who have done research, I myself just finished up my PhD. Research can be a very slow endeavor, and there's a reason it's slow. It should be done thoroughly, and ideas sometimes take time to sort of mature and to come to fruition. But in response to the pandemic, we had to figure out how we can do this very, very quickly without compromising on quality. So at Stanford University, we had a platform called Eterna that we've been utilizing for the last 10 or so years. And what we've done on Eterna is take this RNA design challenge into a video game. And what I mean by that is you can see on the left, the top left screen, we have launched online this video game that allows players to denote and change individual bases of RNA from A to G, C, or U, and then we interactively show them the shape that RNA molecule is taking. And players can design players can submit many designs for any given RNA molecule. They then can vote on each other's molecules to see which designs are most popular when you have communities of people sort of evaluating your design. So unlike graduate school in some way where you often work alone in a laboratory, you might have some lab mates check in on what you're doing, but it's a pretty small group that is checking over your work and also helping you in your design on a day-to-day basis. What we're trying to do is mobilize a large community of tens of thousands of video game players who will check over each other's designs and through checking of each other's work and also innovating and conversations, come up with new hypotheses to test in terms of RNA shapes. And this is where Eterna sort of has its bread and butter. We then take the top voted designs and then we synthesize them in laboratory. So we order the DNA construct that encodes for these RNAs that have been designed. We synthesize them in the house, and at Stanford University, we carry out cutting-edge biochemistry experiments to test the structure, and also in this case, we can functionally test the stability of these RNA molecules. And then the key piece of actually doing experiments, of course, is to then return the results of those experiments. What does the RNA molecule look like? On the bottom left, I'm showing the result of an experiment called SHAPE in RNA molecule structure probing, which tells players This is the molecule you designed, and in yellow depicted are the highly reactive spots on that molecule. So what you would expect is that if it is paired, so if it is touching another base, you would expect it not to be so reactive, so you'd expect it to be blue. So this player got it mostly correct, but there's these certain spots that pop up, this U here and this A here, that seem to not be paired as we hypothesized. So we can do this for thousands of different molecules, give that back to the players, and the players will take that data and then use that to inform their next design. And we can sort of go around this iterative cycle of players hypothesizing and designing and us testing and confirming results that allows us to make online our scientific process and to engage a community of not only professionally trained scientists like myself and others that work in the laboratory, but also video game players who can be tuning in from anywhere and from all over the world to design RNA molecules. I'm going to show a quick video here of what Eterna looks like when you play it, hopefully. Geez, it might not be working, but let me, oh, here. So this is a simple puzzle called glasses, and it's not a real RNA molecule found in nature, but this is a sort of one of the tutorial puzzles that we allow players to engage with early on. Oh, I apologize. Engage with early on to sort of learn how these design rules between base pairing might work out. So we give the player a shape. I apologize. We give the player a shape, for example, that looks like glasses. In its natural form, the RNA is totally unpaired before any bases are changed. And then over time, players realize these design rules, like the fact that U stretches of uracil pair with A's, and then stretches of G's pair with C's. And some players actually abstract this out. So sometimes for them, it's not U's and A's, it's just, yellow and blues pair and G and C, red and greens pair. So over time, players can learn these rules and design the RNA molecules to achieve the desired shape that they would like. So you can see as you start to solve the puzzle, you achieve the shape that we wanted you to, and then it gives you a nice bubbly background, and then it tells you that you completed the puzzle. So you can think about this as the simplest case of how Eterna might work to teach someone how RNA structure works, but over time, they can tackle immensely complex RNA molecules, as I will show on the next screen. So in OpenVaccine, what we try to do is look at the SARS-CoV-2 spike protein mRNA, along with other promising mRNA molecules. And then we sort of look at what standard codon optimization or sort of sequence optimization might do to this molecule. We identify vulnerable spots, and we see that the shape of this molecule isn't quite ideal for stability, thermal stability or kinetic stability. And what we do is we now launch this as a challenge on Eterna to where players are designing not like the simple RNA molecule I showed, but immensely large, complex mRNA molecules, like the one you see here, which is the spike receptor binding domain, which is one of the important proteins for recognition in SARS-CoV-2. So on Eterna, we crowdsource, or we enlist the help of thousands of video game players to help us design these really complex RNAs. In parallel, what we do is we talk with players constantly to ask ourselves, what are some design rules that every player or many players might be successfully employing? And we encode those into computational algorithms for mRNA design. And then after we have these computational algorithms, we often return those results and tools to players in the shape of little tools that you see on the top left. And over time, you can go from a molecule that looks very unpaired, so has a lot of yellow or purplish regions, to this very highly optimized mRNA sequence designed by a longtime Eterna player, a video game player, Eli Fisker, where the sequence of the spike protein mRNA molecule, which is about 4,000 bases in this case, have been exquisitely optimized to feature very few unpaired regions like you see on the right. So a molecule that is mostly blue has been optimized almost base by base by this player over obsessive video game playing, essentially. It is able to be shown to be much more stable in solution than the wild type sequence, as you might see, or what other standard tools might return to you. So outside of COVID-19, I wanted to highlight the different efforts that we have pursued on Eterna. The first is RNA sensors. So we can make different shapes of RNA that bind very important molecules or serve as diagnostics. We then carried out, we're pursuing the building of RNA robotic machines. So RNAs that can carry out calculation as well as human-defined function through complex design of 3D structure coming soon. We are applying to real-world problems such as COVID-19, obviously, in vaccine design, but also in the past we have designed through our project OpenTB diagnostics for tuberculosis that work very simply like a pregnancy test instead of the more complex diagnostic that tuberculosis currently requires. And also we've lent our efforts to a project called OpenCRISPR, CRISPR being one of the most exciting new technologies in science. It was recognized by the Nobel Prize this year to Jennifer Doudna and Emmanuel Charpentier. A gene editing technology that holds a lot of promise, but still needs some work to make sure that it is safe and equitable for everyone who may benefit from the technology. So OpenCRISPR was our contribution to design better guide RNAs to make the technology have sort of like a safe switch. This has been very productive for us. On the right, I'm showing some of the manuscripts that we have been able to contribute to the scientific community. Many of these are preprints and many of these are also peer-reviewed publications. What's notable is this bioRxiv preprint, and we have over 20 of those preprints out now. They're often authored by people not with formal scientific training like myself, but video game players who have learned science to a publishable unit through Eterna by consulting with professional scientists at Stanford University, as well as other institutions that we collaborate with, like Northwestern University in Chicago. So to summarize what we've been doing on Eterna so far, for the OpenVaccine Challenge, we have launched many puzzles. I think we are on our 7th round of puzzles of designing mRNA vaccines. I think we have about 4,000 participants and probably 40 to 50,000 submissions for better design. We've also launched a mobile app to allow better accessibility of Eterna to video game players who may be in parts of the world that don't have readily available access to a computer and web browser. We've been continuing to do experiments in shutdown through a lot of video meetings and scientists going in and taking a lot of precautions to make sure we can functionally characterize these molecules. And our hope is that our findings will be integrated in an open and equitable way towards these molecules that are going into clinical trials, so that the vaccine can be widely distributed fairly through the findings of citizen scientist efforts. And then we have some preprints that are coming out. I believe we have one already out, but we have a couple of really big ones, sort of summarizing our findings and communicating to the world what we've done. So please stay tuned for those. I would not be correct if I said this was a isolated effort. This is an effort of many people working tirelessly, members of the DOS lab at Stanford, vaccine developers, meaning people who are working on the background to make improvements on the technology side to make sure that video game players have the tools they need. They work with us to do media, a lot of work on the back end to make sure the platform can run smoothly. People in the Barna lab at Stanford University who have been carrying out a lot of the critical experiments in cells to make sure these mRNA molecules work as expected. Professor Pasupanyawala Shu at Stanford University, and all the staff at Stanford who've given extra time to make sure we can be successful in our efforts. A lot of collaborators, including PBS, which is a large network in the US, as well as other collaborators at Stanford University and other institutions. We currently have a collaboration with King Abdulaziz University in Saudi Arabia, as well as Carnegie Mellon and other institutions. And of course, there's been tremendous support, financial support from many different donors that have made this project possible. And then finally, I want to give the most thanks to the Eterna video game players. This is a snapshot of some of our longest running and most active players who have participated in full for this challenge. And it's thanks to their efforts that we were able to carry out this magnificent effort in redesigning mRNA vaccines in an equitable way. Finally, if you would like to be involved, please feel free to reach out to me personally, but also you can just navigate to eternagame.org. You can register for an account in less than 5 minutes. We have a YouTube channel and Twitter, all the standard social media channels, but I would urge you to go to eternagame.org, play a couple of the introductory puzzles and see what it's all about. And it's quite exciting. And it's been really great to be able to share our work with you today. Thank you.
Thank you, Dusun Kim. I hope you stay still with me. I can't see you, but can you hear me?
Yes, I can hear you.
Yeah,
can
great.
you hear me okay?
So I will just guide you a little bit through the maybe left 7 minutes, so we could maybe get some questions from people taking part in this session. So please, if you want, just write in the chat or raise your hand. And maybe in between, I would like to ask some questions or maybe ask you, could you maybe shortly explain, so maybe some of the listeners know that in principle the order of the bases on the RNA translates finally into the sequence of the protein. So why is it possible that you can change this? So maybe you can shortly comment on what codon optimization in principle is.
Oh, right. So for mRNA molecules, the end product, that is a protein, are encoded by these triplets of bases. So there's, ACG will code for an amino acid, GCU will code for another amino acid. But thankfully, there's this property called codon redundancy, which means there's about 20 naturally occurring amino acids, but because there are triplets of ACG or U, there's actually 64 possible codons. And out of the 64, 3 are stop codons. So there's about 60 or so 61 codons that code for 20 amino acids, which means for any given one amino acid, there should be about 3 different codons that can encode for that amino acid. So because you can code for the same amino acid with 3 different codons, at each amino acid, which are the building blocks of these proteins, you actually have 3 or so options of which codon you will choose. So you can't freely mutate everything everywhere, but there are these positions of flexibility where changing an A to a C will change the RNA sequence, but it won't change the protein sequence. So our players, through the tools that we've given them, so if you go on Eterna and do a challenge, it'll highlight for you the places where you can change, and it'll also keep track what the amino acid sequence is as you mutate this RNA. So thanks to the fact that there are more codons than amino acids, players can pick and choose which codons they will use to flip or change the order of the bases in the RNA molecules.
Okay, and maybe one short other questions we got was, what are the advantages of mRNA vaccines compared to maybe other variants of vaccines? If that's short to answer.
Yeah, so I think the biggest advantage that mRNA vaccines have shown and hopefully will show is the speed to which we can make this. So the reason mRNA, so as a testament, in the US at least, the first mRNA phase 1 vaccine for COVID-19 went into a patient about 60 days after we had the sequence of the coronavirus genome.
Wow, that's really fast.
And this is because once you know the genomic sequence and the protein you're encoding for, you can sort of manipulate the mRNA molecule like you would computer software to encode for a different molecule. With protein vaccines, typically you have to think about how the protein will be made and manufactured, and there are challenges in doing that very quickly. So I think the biggest advantage of mRNA vaccines is the speed and flexibility. But I should say there's also many challenges. So they're like the one we're addressing. So it's not this perfect technology that should be used in everything, but in responses to pandemics like the one we're going through, it's a very advantageous characteristic to be so fast to respond and to be flexible.
I also have a put up hand by Victoria. I will try to unmute her so she can put her question herself. We have 2 minutes left, Victoria. Please.
So my question is 60 days, you said. Am I right?
60 codons, yeah, so 60 sets of...
How many days from the sequence to the first injected RNA vaccine?
Oh, days, yes, 60 days.
I'm now wondering if the administration is that fast that you in 60 days got the all the protocols that you're allowed to test it on the people.
Yeah, it's difficult to answer. I'm not sure.
I suppose.
I mean, we can make it maybe even faster, but we have to think about also about this part, this side of the whole story, that we need these concrete protocols, administration regulation, I don't know what else, to be allowed to test it. And
Victoria, I think
days is enough.
Victoria, I would think that this is for the very first, so not for a broad population, so where you need a lot of administration, but I think self-testing might be allowed in a very early stage, and that's maybe what was done. But unfortunately, we cannot really continue the discussion now because our time is already gone. But it was a very interesting session, and I liked it very much to see what is possible with people that are not coming from science and can do that in computer games. That was really very good. Yeah, we would have some last question maybe open. Maybe I can put this very last one. The question was, are players better than algorithms?
For many of the most complex ones, we have consistently seen humans are better than computers so far.
Okay, thank you for this quick answer. So thank you very much for joining us. And we will now move